Very good day, my dear postgraduate students. Thank you for the 1000th class completed by R.A.T.S. The topic for the day is telomeres in health and disease. Think of the subheadings. The subheadings will take you to the answer. And structurally, we are going to talk about the telomeres here. And there is a centromere, the short arm and the long arm, which are being capped by the telomeres. So it is, in fact, a short nucleotide sequence that repeatedly occurs at the ends of the chromosomes in eukaryotes. Telos means end and meros means part. And on the screen, we are seeing the famous personalities who had been awarded the Nobel Prize. And obviously, when we are going through the panel of utilities of these, we shall know how well they are being justified. There is a repetitive nucleotide sequence, TTA-GGG, that occurs 2500 times repeat. And therefore, a loss of this may cause the cellular aging. So this is an important diagram and a text for us to follow. The telomeres consist of repetitive DNA sequences. Look at this one, TTA-GGG. And it is being complemented by means of CCC-TAA. So there is a primary one and then a complement of it. These two together form the double strands of it. And the telomere end has got a cap which might not be virtually affecting the chromosome, but literally protects it. There is a shortening of it with repetitive cell divisions, as a result of which at one point of time it becomes critically short and the chromosome suffers. Therefore, it can be considered to be a mitotic plaque. So, this is from current opinion pharmacology. And this is the cap that we have been talking about. So, this is the T loop that is over here, which will be covering. And we shall be telling you the importance of it. This per se does not participate in the chromosomal activity as such, but rather protects it. So I would like you people to kindly explain this. Diagrammatic picture. So you find that there is the telomere at the end of the chromosome. So there are 4 telomeres over here. And there is a shortening of the telomere after the cell division. Multiple replication, there is a progressive shortening of it. At the end of it, in old age or senescence, you find that the telomere, at the end of which the telomere is shortened and there is an exposure of the underlying chromosome. Telomerase, on the contrary, helps to build this particular cap protecting the chromosome. Probably there will be immortality in some conditions. This we shall be seeing in our text. So this is a sequence we have already touched upon. I will not be going into it. A very important line diagram for you to recapitulate all the material. The asterisk mark here indicates that it is a very important text, kindly by heart. It forms a vital process of multiple cellular divisions. And it protects the chromosomes. And what happens is, as long as this is there, there is no end-to-end fusion or chromosome-to-chromosome fusion. Probably the Robertsonian ring chromosome, etc., might be because of this. And there is a binding called the sheltering. So there is the DNA and then there is a repetitive one, and this is being covered by the sheltering proteins, which are very important. And they are involved in the chromatin remodeling and protects the ends of the chromosomes. There are 2 key telomere features. One is the T loop. That is what we had shown in the previous diagram. And the other one is transcription of the telomeres. pi tera. Telomeric repeat containing RNA. That is why it is called as tera. Tera molecules are crucial in the regulation of the telomerase activity as well as in the formation of the heterochromatin at the chromosome ends. This text will appear a little complex, but then once you people rehearse repeating it, it will be easy for you. R.A.T.S to face the exam. So during each of the cycle, you find that there is a shortening of the telomere by this particular unit. Normally it is about 50,000. So a small amount will be shortened. And the DNA polymerase is unable to completely replicate. The G-rich telomere sequence is susceptible to the DNA sequence. Senescence means in old age, the telomeres are being completely damaged. This can be one of the causes is oxidative stress. So this is it over here. There is a G strand and then a C strand. They together form a loop. And this particular loop protects the end of the chromosome. So what is the importance of the telomere? It protects the chromosome, we had said so far. And we had thought about aging. Now what happens when your vehicle is running? It can be having a complete tank or progressively as it runs. There is an emptying of the fuel, there can be a near empty tank. That is what is happening in aging. So these are the things we had seen, separation of the chromosome. Without this, the chromosomes also cannot be repaired. And they form the cap. And it is analogous to the plastic at the end of a shoelace. Look at this. This is probably a superb explanation that has been given. And then there is a plastic cover over here. Without this, obviously, you people will know what damage will be there to the shoelace. So telomerase is what we had seen. It stabilizes the length of the telomere and it replicates this particular unit. And there are two enzymes basically. One is the TERT, telomerase reverse transcriptase, and also the TRA-TERC. The next one is discarate. And these are being coded by means of the TERT gene or H-TERT as it is called. They form a wrap around the DNA. Now, what is the function of the telomerase? Somatic cells do not have this, therefore they have got a limited lifespan. Whereas a high telomerase activity is seen in cells which are constantly multiplying. They can be the germ cells, the stem cells, the epidermal or the mucosal cells, hematopoietic stem cells, cancer cells. And it can be either switched on or switched off. When it is switched off, the cell dies, and when it is switched on, it continuously multiplies. That is the secret of the cancer. An anti-telomerase is used for the treatment of cancer. Obviously, you people can explain. And when I say stem cell, it is by itself a question. We shall be going into it. It can be a non-committed or a committed stem cell. What is the significance of the telomeres and the telomerase? So the application of it. What is the topic as such? Telomeres in health and disease. So in immunology, there can be an immunodeficiency, metabolic disorders. So the gene being affected, there can be obesity, diabetes mellitus, the second type of diabetes, cardiovascular, hypertension, endocrine, polycystic ovarian disease, which again is a multigenetic disorder. Cushing's syndrome, acromegaly, aging. It can be cellular senescence or other age-related diseases. Somatic cells, no telomerase, immortal cells have got innumerable or unaccountable telomerase. Liver, non-alcoholic fatty liver disease, viral hepatitis and cirrhosis. These we do not know from where it comes. Once you go into the ocean of telomeres and telomerase, you find that there is a never-ending plethora of topics which we can delve into. CNS, that can be the Alzheimer's, oxidative stress, defective DNA repair. And it continues over here, metabolic syndrome, decrease in HDL, increase in waist circumference. triglycerides, fasting glucose. So obviously they are dealt with in diabetes. Proteins, SAS. This is important. You'll have to make a mention of it. Senescence associated secretory protein. And TNF and interleukin are all related to it. Insulin is one such thing. Cellular aging is over here, and mucocutaneous. This is an important syndrome I'll be touching upon, the leukoplakia, nail dystrophy, and pigmentation. Psychiatric disorders also come into it, and the treatment for the various age-related disorders. This is again, I would like you people to kindly explain. So we are being exposed to a lot of these stresses, oxidative, metabolic, etc., hormonal, which can lead to the telomere shortening. Once there is a telomere shortening, there is a cellular senescence. We had mentioned this one earlier. So this can lead to a number of these metabolic or endocrine disorders, which can further lead to telomere shortening. So it becomes a vicious cycle. And this particular pathway can block it, P53. But this again is being cut when there is going to be an absence of telomerase. So this has to be a therapeutic target, both for cancer as well as for targeted therapy. So once again, the telomere shortening, what are all the various causes for it? We have mentioned over here. And these are again a number of diseases over here under the metabolic and the endocrine topic. Kindly go through. So there is a paradigm in cancer. Telomere loss, it can lead to a limited lifespan and it induces genomic instability. But when there is a telomerase activation, it protects the chromosome ends. We thought it was protective, but then it promotes immortalization, as a result of which it promotes cancer. And this is a wonderful article for reference. You people can go to it. The lengthening and shortening of it, again, physiological activity, you can divide it into like this. It can be physiological, it can be oncogenesis. Under physiological, you have got the stem cell, organ homeostasis, immune modulation, and age process regulation. Whereas oncogenesis is the cancer development, progression, metastasis, as well as resistance, which means refractory to treatment. This is again a beautiful tabular column, which I want you people to reproduce. So this again over here, there is a shortening, there is a loss of function, there can be an arrest of the cell cycle, and there is a death of a cell. Or sometimes there can be a programmed death such as apoptosis, senescence, genomic instability. And there is an expansion of the abbreviation for this. Once again, it is a recollection table. I would like you to go through the various organs of the tissues and find out what is it. There are certain things are important for us, such as leukoplakia, hyperpigmentation, nail dystrophy, and cirrhosis. See over here in aging, there is a telomere shortening, increased apoptosis, inflammation, tissue dysfunction. Also, there is a decreased telomere expression, increased mitochondrial dysfunction, oxidative stress, again leading to tissue dysfunction. This probably would have been given in one of the first chapters in Robbins. Therefore, a knowledge of this kind will help us understanding the first chapters. This is what I had been saying. Look at this one. There is a leukoplakia, and then there is a cutaneous pigmentation over here, dystrophy of the nails. And this is a particular syndrome that is there. Others are Bloom syndrome, Fanconi's anemia, and Niemann-Pick break syndrome. So these things you people can mention. The classical thing is Werner. Though we all know it, I was very much astounded to see the features of it. The classical one being the long beak-like nose. So found out by Werner over here, 1 in a lakh, and located in chromosome 8. Adolescent, there is a spurt of growth, and there is an alopecia, obviously. There can be a wrinkling of the face that is there, beaking of the nose, as I had mentioned. All of them have got this one. Cataract can be there, atherosclerosis can be there, and this is a mode of inheritance of it. Thanks to again, hereditary cancer in clinical practice. These are all the various features given in a diagrammatic way, which again I would like you people to go through. See in the bone marrow what happens. There can be a cytopenia, dysplasia, aplastic anemia. Or in the lung, there can be a fibrosis and an emphysema. Liver, obviously, there is a fibrosis and a cirrhosis. Mucocutaneous, we have already seen. And there can be an alopecia also. Cancers, various cancers have been linked to a defect in the telomerase action. And once again, nature. These are all superb ones. They will appear like very simple diagrams, but then the text is fantastic. And you find that there can be an AMD, okay, in the eye. What is AMD? Please find out. In the brain, there can be an Alzheimer's disease, Parkinson's disease again. You find in the lung, these are all the features. So there can be a macular dystrophy or dysfunction. This is a very important diagram which I would like you people to kindly understand. As a student writing the examination, this will be the key picture for you people to write. So you find that there is a squash of it that is being prepared so that you have the DNA extract. So, and the cells, they are being arrested at this particular place. Usually we use this colchicine when we want to study the G banding technique. You'll be finding in the chromosomes, there will be the alternate dark and the light bands. They are called as the G bands or the GMSA bands because they are stained and demonstrated with GMSA. But in this particular one, you find that the cells are being transferred to, I have given it in a box. I would like you to kindly by heart it. I have put hypotonic solution, and then they are fixed in methanol glacial acetic acid. Again, there is a second fixation in formaldehyde. There is treatment with pepsin, and ultimately there is a hybridization over here. And you find that there is a complementary sequence that is being formed, and finally you stain it with DAPI. So this is what is happening, and the image is being captured because these are all fluorescent, and fluorescent means there is a time limit. Therefore, you will have to take a picture of it. And what happens, thanks to Wikipedia for this particular diagram, this is exactly. So you find, are you able to see the banding over here? I'm able to see the dark and light bands, but this is not sufficient for me. But when I do the fluorescence, I'm able to see that there are some cells which are having the yellow ends of it, whereas the others do not have. Therefore, the intensity of the yellow end tells us what is the activity of each telomere. So that is the principle of it. This I would like you people to kindly explain in detail. And the same diagram is over here, not for repetition. What are the other methods? It can be a Q-fish, which is quantitative. You can assess. Flow fish, it measures the signal, but you cannot say the number of cells. And then there can be a comparison between the normal population and the subpopulation of cells. Between the neoplasia and the surrounding tissue, you will be able to study. Therefore, a targeted therapy is possible thanks to this wonderful diagram and reference from Wikipedia. I'll never be an old man. To me, old age is always 15 years older than I am. That means I am younger than what I should be by 15 years. Beautiful sayings by Francis Bacon. So kindly preserve this one and forget my class. Thank you.